it turns out that we have here a drug that's unique in that it has two activities, one against the AIDS virus and one against the fungi that infect individuals who have AIDS. And this opens up the door to the possibility that this will be an excellent treatment at low cost for the third world.
We think that the amphotericin monoester or amphotericin methyl ester shows tremendous promise, and we hope to be able to follow this project up with a larger study in a more relevant species, perhaps in non-human primates.
We feel that the strong potency that AME shows in culture really strongly argues that clinical trials should be carried forth with this compound to assess whether it serves not only as a very potent antifungal agent, but also as an anti-HIV compound. We strongly feel that these studies should be pursued.
This project definitely is going to have a great impact on Africa. The uniqueness is that it is a medical solution for Africa and the third world.
My name is Glenn McGill. I'm with Rock of Africa Mission, an interdenominational Christian mission based in sub-Saharan Africa. I'd like to introduce you to a most exciting project that could bring enormous hope to people in Africa and throughout the third world. I know firsthand the devastation that AIDS has brought to Africa. I have seen and even felt the hopelessness that victims feel. After sharing about this tragic situation recently in New Jersey, a group of women began praying for the discovery of a low-cost medicine that might treat the countless numbers of people in Africa out of reach of expensive HIV medicine. Their passion and their numbers grew, and from this has become a most exciting project. During the 1970s, a medicine to treat aggressive forms of infection called AME was developed by Dr. Carl Schaffner and other scientists at the Waksman Institute of Rutgers University. Patents for AME were secured and the research began in earnest. This project, however, was abandoned before its completion, long before the arrival of the AIDS pandemic. Now, some 30 years later, the patents for AME have expired. Dr. Schaffner and several other researchers have carefully re-examined the AME research and now confirmed that the project was stopped because of errors in production unrelated to AME itself. In fact, recent testing has now revealed that in addition to fighting infection, AME attacks the very HIV virus itself. While this is indeed great news, in a research industry sustained by a financial incentive, AME, now without any patent protection, has very little value. However, because patent-free competitive manufacture is the very key to bringing down any medicine's cost, AME shows great promise to bring a low-cost medicine to treat HIV in the third world. Please take a moment to listen to Dr. Schaffner and others about this exciting research, and then consider how you might join us in a remarkable opportunity to bring low-cost medicine to treat HIV in the third world.
The discovery of streptomycin by Dr. Waksman and Dr. Albert Schatz was a monumental event in human medicine. I had, of course, known about Professor Waksman, and he was well known. I joined him in 1953, 1 year after he received the Nobel Prize for his discovery of streptomycin. And that's how I came to the Waksman Institute. So I spent 45 years at the Waksman Institute as an active professor. I'm now Professor Emeritus, and I've spent all the years since my retirement in 1999 still active in chemotherapy. I had a particular interest in a group of antibiotics called polyene macrolides. And among them is a compound that was originally discovered at Squibb for the treatment of systemic fungal diseases, which was, in one case, was Amphotericin B, which is a rather potent antifungal with tremendous toxicity. In fact, its use is considered a gold standard in treatment because there was nothing better to treat patients with fungal disease. Gradually, other drugs came into the picture, but these, to this day, are not constantly effective in the treatment of fungal disease because of resistance, whereas Amphotericin B doesn't ordinarily develop resistance. And it was for this that I started the structural studies of this compound. And in the course of these structural studies, my group prepared a number of different derivatives for X-ray crystal analysis. And one of them turned out to be the methyl ester of Amphotericin B. We then had a collaborating clinician in University of California, Davis campus, who was interested in valley fever. And he had animal models for valley fever. And he got a sample from me of this Amphotericin B methyl ester, which we shortened as AME. And he found that it was very effective in treating coccidioides infection or valley fever in these mice, whereas the Ampho B, the parent compound, which was already in clinical use, was much too toxic, and they would die of the drug before they were cured of the fungal disease. This, of course, excited him tremendously, and he decided to file for a compassionate IND from the Food and Drug Administration. And he proceeded then with a few patients who were considered terminal with valley fever. They had disseminated disease which involved their brain, their central nervous system, and they were rather in poor shape. They had already been on a number of antifungal antibiotics, including Amphotericin B, and Amphotericin B had compromised their kidney to the point where they had to stop taking it. They received this derivative, AME, and they were cured. And their kidney function was restored in spite of the fact that they were getting high levels of this new derivative. Of course, this excited everybody at the University of California, and of course, the company that made the material by our process for this trial. Thereafter, there was a dust storm in California, in San Joaquin Valley, and apparently this spread so wide that many people came down with valley fever. And there were over 50 patients with disseminated disease that entered into the study. It was enlarged, and the company then provided the material. But what happened was these high doses of this AME that they received was gradually associated with some neurological toxicity. And when they investigated this toxicity, found out that it was there was cranial damage, there was white matter degeneration of the brain, and everything stopped. And nobody bothered to explain what went wrong. Our very first 5 patients that we had didn't show any of these neurological toxicities. And of course, the Food and Drug Administration stopped the trials, and we did no more with this drug in the clinic. It turned out that I received samples of the materials that had been provided by the company for this trial. And we had developed a method of high-pressure liquid chromatography at that time for these compounds, for this sort of compound. It was, up to that point, very difficult really to analyze these preparations of these so-called polyene macrolides. And we employed these techniques, and we found that the material that was initially used and provided by the company was essentially AME, whereas all of the lots that were used where the patients received high doses over an extended period of time, and the appearance of the neurotoxicity, these lots were all complex mixtures. And of course, this made me very suspicious as to what component of this mixture was involved with this toxicity. But at that time, we could get no funds to do any further work with this compound. And so I had to stop research with this. It was only later that the HIV epidemic came into being, and I realized from previous studies with other lipid-coated viruses that it should have a potential for HIV. So I contacted Dr. Robert Gallo at the National Cancer Institute in Bethesda, and he tested this AME against the HIV virus and found it to be very active in inhibiting the infectivity process of the virus. And this was all done in cell culture. And then many other studies followed in collaboration with the Department of Health of New Jersey in Trenton. And we had a student from this institute who worked in the facility for HIV at this Department of Health facility in Trenton, New Jersey. And we found out that this compound affects the infectivity process directly by interaction with the virus, rendering the virus non-infective, directly with the host cells that are infected by the virus, rendering them immune to infection. So it had a rather very unique mechanism of action as compared to some of the other antivirals like AZT. We also discovered it was synergistic with AZT, making AZT more active than by using AZT alone. Well, I then thought that this was important enough to do some work. I had a collaboration at the School of Pharmacy with the Department of Neurotoxicology. Since the alleged neurotoxicity of AME was in question, and I had some serious doubts as to the composition that was used in this ill-fated clinical trial, I contacted Dr. Kenneth Rule, who is an eminent neurotoxicologist, and he became interested in the project. And we then proceeded to test AME with some of the components of this mixture that had been provided by the company to these patients who came down with neurotoxicity.
About 12 years ago, Dr. Carl Schaffner, who is at the Waxman Institute, contacted me about my willingness to participate in studies of a novel antibiotic chemical, Amphotericin monoester, which he had developed. I'm a neuropathologist by training, and I have a great interest in pharmaceutical neurochemistry and neuropathology. And so I agreed to meet with Dr. Schaffner and discuss the potential testing of this chemical. Dr. Schaffner at that time filled me in on the history of the chemical, that it had shown tremendous efficacy in the treatment of terminal fungal diseases in humans, and some of the problems that had occurred in the treatment of patients, specifically the development of white matter demyelination as a consequence of contaminated chemical being used in the treatment of patients. Dr. Schaffner is one of the few people in the world who could produce pure amphotericin monoester and made it available to me for studies of the neurotoxicity of this chemical and its contaminant, Amphotericin B, and the most common antifungal agent used currently, which is Amphotericin B. So we could look at the pure chemical toxicity, the mixture, contaminant toxicology, and then the potential neurotoxicology of the Amphotericin B. We did the study in rodents, which is the primary first-line testing species. We used rats, and in combination with an undergraduate honors student from the School of Pharmacy by the name of Manisha Vapawala, we performed a chronic study of Amphotericin B plus AME monoester and the pure monoester, and then did a thorough clinical chemistry and histopathological assessment of the brain. Much to our pleasure, we very clearly demonstrated that it was the Amphotericin B contaminant in the chemical that was used to treat the patients, and not the amphotericin monoester itself that was the neurotoxic agent. Indeed, we were incapable of producing true neurotoxicology with the pure amphotericin E at any concentration that we used. And we used concentrations that were in excess of what would be used clinically, even on a compassionate IND for terminal patients. Our conclusion based on this study, which was a fairly extensive study, was that the neurotoxicity observed in the patients was almost certainly the consequence of using impure, contaminated compound from the company, and that it would perhaps be unlikely or less likely that white matter demyelination, neurotoxicity, would occur with the pure chemical.
In more recent times, I had seen some publications by Dr. Eric Fried, who's chief of the virus cell interaction section of the National Cancer Institute's division of HIV drug resistance program. And he became intrigued with AME as a model compound for infectivity studies, and he proceeded to test the materials I sent him, and within a very short time he was writing abstracts.
We've been studying in the lab for several years the ability of cholesterol depleting agents and cholesterol disrupting agents to inhibit HIV-1 replication. One of the compounds that we've been studying most recently is Amphotericin B methyl ester, or AME, which in culture shows very potent antiviral activity. As you certainly know, AME has been used previously in clinical trials with mixed results, apparently because of contamination with Amphotericin B, the parent compound from which AME is derived. We feel that the strong potency that AME shows in culture really strongly argues that clinical trials should be carried forth with this compound to assess whether it serves not only as a very potent antifungal agent, but also as an anti-HIV compound. We strongly feel that these studies should be pursued.
The compound AME is no longer under patent. This patent has expired some years ago, and no commercial source company would want to undertake studies with this when there's no patent protection, and it would be open to generic competition immediately. And so as a result, no one is willing to pick up the ball and say, hey, we should put this in patients, we should test this, we should do some more toxicology, and proceed with developing this as a drug for the treatment of AIDS, as well as for the treatment of fungal disease in AIDS patients. It turns out that we have here a drug that's unique in that it has two activities, one against the AIDS virus and one against the fungi that infect individuals who have AIDS. And this opens up the door to the possibility that this will be an excellent treatment at low cost for the third world. This is something that has really driven me to proceed and do what I can to try to get a clinical trial underway. The drug has never been given to an AIDS patient. All of the patients in the past studies were free of HIV, because at that time it wasn't as well in position as it is today. This is a pandemic of the highest order. And at that time, in the 1970s, there was not even a recognition of AIDS as an emerging disease. So that we have already some indications from one study which was run in Louisiana in a facility there where they used Amphotericin B for treatment of fungal disease of AIDS patients. It turns out that they were monitoring the viremia, the virus titers in these patients in the short course of treatment, because the toxicity, the kidney damage that's associated with Amphotericin B treatment prohibited continuation of the study. So, but they did find that the viremia was significantly decreased as a result of giving them Amphotericin B. With AME and the rapid excretion and the less toxicity, this will permit a longer-term treatment to see what effect AME has on the viremia, and I anticipate it will be active against that disease in this way, that you would decrease the titers of virus in the blood. We know from studies here at the Waksman Institute that adding AME to blood contaminated with AIDS virus renders the virus inactive. So that in blood, the compound does have a direct effect on the virus. And that is very important. So with the lack of a patent, if this could be made after it's proven that what I'm saying about the effect on the virus infections, we know it's highly active against fungal disease, I think this would be a remarkable God-given treatment for the third world and the poor individuals that are suffering there with AIDS and with fungal disease. It was through Dr. Robert Parsons of the Hayden Watson Foundation that I had the occasion to meet Glenn McGill of the Rock of Africa mission, and he has taken interest in the possibility that we could treat patients in Africa with this drug once it becomes available, and this of course will take money. And he has gone through several countries of Africa, and he's finally now made arrangements with enthusiasm on the part of the clinicians in the country of Ghana at the KNUST, which is one of the major universities in Africa. And these clinicians are anxious to start a study, and they will start conservatively, to ascertain the toxicity of this, as well as the efficacy on fungal disease and on the virus titers of these AIDS patients. So that this is a unique opportunity, and I must say that Glenn McGill has done a tremendous job in getting to a point where somebody is actually anxious and enthusiastic about evaluating this drug further, in this time in AIDS patients. I feel that this opportunity now in Ghana is a monumental opportunity for us to proceed with a drug that could help the third world.
The Kwame Nkrumah University of Science and Technology, Kumasi, which I represent, is very excited about this AME project. The president of the university, Dr. Kwesi Andam, is very committed and is ready to provide all the support that the project actually needs. This project definitely is going to have a great impact on Africa. The uniqueness is that it is a medical solution for Africa and the third world.
So this is something that could be administered over a long period of time at lower doses, because they find that as little as 1 microgram per milliliter is sufficient to inhibit the virus infectivity. And the levels that have been obtained in the past in primates as well as in humans is 10 to 20 times higher, just with low doses of AME by intravenous parenteral route. Another thing that this brings to mind is that in the original clinical trials with over 50 patients, they found that Amphotericin B had to be administered by intravenous drip for a period of 6 to 8 hours, and the patient had to be hospitalized. When they were treating patients with AME, they found that the time of administration could be shortened to the point where an intravenous bolus of the drug could be given directly without a drip procedure. This is, of course, tremendously important in the third world because you can't put all these people in hospitals. You could treat them all as outpatients, and this is a tremendous advantage in the therapy of a large number of people. This work with AME in the clinic would have tremendous implications in treatment of fungal disease and HIV infections in the whole world. The effects are rather immediate. We don't have to wait 2 years. We would see an effect within a week or less. And this is what is so important, that we can be able to provide funds and give us the opportunity to proceed with this rather remarkable drug.